Hello everyone, my name is Saba Ghaffari. I'm a computer science PhD student at the University of Illinois at Urbana-Champaign, and I'm going to talk about my project on integrated analysis of multi-omics data to identify transcriptional regulators of cancer progression. More specifically, in this project, we are interested in finding about the transcriptional regulatory mechanisms underlying colorectal cancer progression, as is the second leading cause of cancer-related deaths globally. For this purpose, our collaborators in Mayo Clinic provided us with the multi-omics data measured on an invasive subculture of SW480 cells that was established by repeated selection of cells that could invade through a porous membrane coated with metrogel. The measured multi-omics data include gene expression, histone modification for 4 different histone marks, and DNA accessibility in 4 stages of colorectal cancer invasiveness. But in this project, we only use the data from the earliest stage and the latest stage, as well as the TF binding data from ENCODE HCT116 cell line for 20 TFs. And the goal is to identify the transcription factors associated with colorectal cancer invasiveness. Also, I should mention that in the interest of time, I won't talk about the analysis performed using accessibility data. Now let's see what is the framework of our analysis. To find the transcription factors associated with colorectal cancer invasiveness, we aggregate the p-value of differentially expressed genes upon progression with the evidence of TF's regulatory effect on genes using a probabilistic graphical model that enables us to score each TF based on its relevance to CRC invasiveness and also assign a score to each TF gene pair representing the extent to which the gene mediates the effect of TF on colorectal cancer progression. Now let's see how we define the TF's regulatory evidence go for each gene. Going from early to latest stage, we consider the appearance or the disappearance of all histone marks within transcription factor binding site near the gene as the TF's regulatory evidence. Such evidence is encoded with a binary vector for all the TFs for each gene, as shown in this matrix. As for the model, we built upon a PGME model by Casey Hansen to work with multiple TFs simultaneously. You can read more about this model in the original paper published by Genome Research on finding transcription factor drug associations. Now, let's take a look at the relevance of the 20 TFs in this study to colorectal cancer progression. As we perform 2 separate analyses for downregulated and upregulated genes, we have 2 rankings, as shown here. On the left, the ranking of TFs affecting progression through downregulated genes, and on the right, the ranking of TFs affecting progression through upregulated genes are represented. Among the top 5 TFs, JUND is a potential component of the dimeric AP1 transcription factor complex that is generally considered oncogenic. There is also considerable literature evidence for the other highly ranked TFs' involvement in cancer-related processes, including colorectal cancer. We also assessed the value of dynamic epigenomic data. To do this, we repeated the model training using only the presence of a transcription factor binding site as regulatory evidence. As shown here in the rankings, you could see that JUND is no longer a top candidate, whereas it was a top candidate using dynamic epigenomic data. We also took other approaches to assess the value of dynamic epigenomic data that I will skip here in the interest of time. Based on various evaluations, we decided to experimentally validate the association of JUND to CRC or colorectal cancer invasiveness as indicated in the plots. You can see that the knockdown of JUND, represented by green line, affects the invasion and migration of SW480 cells compared with the control case in red line. In addition to the TFs, to ranking the TFs based on their relevance to colorectal cancer invasiveness, we also found a set of 70 genes mediating the effect of the top TFs on colorectal cancer progression, and use them to cluster the TCGA CRC patients using their gene expression, microRNA expression, and somatic mutation profiles. As you can see here, the log-rank test p-value of the survival analysis shows a statistically significant difference in survival characteristics of patients' clusters. And to summarize, we took a comprehensive multi-omics approach to investigate the cis-regulatory mechanisms underlying CRC invasiveness in a well-controlled experimental setting, and identified numerous TFs and downstream targets known to be involved in metastasis-related processes, as well as additional factors that have not yet been directly studied in the context of tumor progression. And at the end, I would like to acknowledge all the individuals who contributed to this work, and NIH, Mayo Clinic, Illinois Alliance, and Compton Initiative for funding this work. Thank you.